protein binding. Protein binding is a specific, often reversible interaction between a protein and another molecule called a ligand. This interaction is fundamental to nearly every biological process. What is meant by protein? Larger molecules that provides the binding site means enzyme, receptor, antibody, or carrier protein like albumin, ligand, molecule that binds to protein. This can be small and large. Small means hormone, ion, drug, substrate, or large that is means protein. Characteristic of protein binding specificity: the protein's binding site is structurally and chemically complementary to its specific ligand. A classic example is a lock and key model. Example: enzyme hexokinase binds glucose but not other similar sugars like galactose. Affinity: the strength of the interaction between a single binding site on the protein and its ligand. His high affinity means the binding is strong and complex is stable. Reversibility: most of biological binding is non-covalent and reversible. The ligand bind and then dissociate, allowing for rapid regulation. Protein plus ligand that gives a protein ligand complex. Structurability: since a protein has a finite number of binding sites, the binding can be saturated. Once all sites are occupied, adding more ligand will not increase binding. The force involved in binding: the interaction is stabilized by multiple weak non-covalent forces. Hydrogen bonds between uh, that means between polar groups. Electrostatic interaction that means between positively and negatively charged amino acid side chains. Van der Waals forces. Weak attractions between closely approaching atoms. Hydrophobic interaction, that means the tendency of nonpolar groups to associate in an aqueous environment, excluding water. Types of protein binding with example: type of binding, protein example, ligand example, biological consequence. Type of binding: catalytic, that means enzymes. Protein example, hexokinase. Ligand example, glucose and ATP. Biological sequence: catalysis, convert substrate, that means glucose that is converted into a glucose 6-phosphate. Structural protein example, tubulin. Ligand example, other tubulin proteins. As biological sequence, assembly from microtubules, a key part of cytoskeleton. Type of binding, transport. Protein example, hemoglobin. Ligand example, oxygen. Biological sequence, carrying, that means transport oxygen from lungs to tissues. Then type of binding, transport. Protein example, serum albumin. Ligand example, fatty acids and drugs like warfarin. Biological sequence, solubilization and storage, carries insoluble molecules in bloodstream. Fourth one, signal transduction, that means receptor. Insulin receptor, insulin signal transmission, binding triggers intracellular signals for glucose uptake. Immune response, insulin receptor example, antibodies, that means immunoglobulin. Insulin antigens, that means virus particles. Signal transmission, binding triggers intracellular signals for glucose uptake. Recognition and neutralization, identifies and marks foreign invaders for destruction. 6. Gene regulation, transcription factors that means P53, insulin, DNA, that means gene expression control, binds to specific DNA sequence to turn genes on or off. The binding site, specific region on the protein where the ligand binds. What's the characteristic means? Formed by a 3D arrangement of amino acid side chains, often a cleft or cavity on the protein's surface. Then its chemical properties like charge, hydrophobicity complement the ligand. Allosteric site, a special type of binding site that is distinct from the active site. Binding of ligand here causes a conformational change in the protein that alters its activity at the main site. This is a key mechanism for feedback of inhibition. Biological and clinical significance, regulation of metabolism, feedback inhibition, where an end product binds to and inhibit an early enzyme in a pathway. Cell signaling, hormones and neurotransmitters exert their effects by binding to specific receptor. Oxygen transport, cooperative binding in hemoglobin allows for efficient oxygen loading and unloading. Drug action, most pharmaceuticals work by binding to specific protein targets, that receptor and enzymes. Agonist, bind and activate the receptor, that means morphine binding opioid receptors. Antagonist, bind and block the receptor, pre- preventing activation, that means beta blockers. Pharmacokinetics, the binding of drugs to plasma proteins like albumin affects their distribution, half-life and efficacy. Only unbound free drug is active. Significance of protein binding, drug action and pharmacology, drug efficacy, only unbound drug or biologically active, drug duration, that means protein binding creates reservoir, prolonging drug action, drug interaction, competition for binding sites can increase free drug levels. Dose considerations, high protein bound drugs require careful physiological regulation, hormone action, enables cellular response to signal, example insulin binding. Enzyme control regulates metabolic pathway via feedback inhibition, oxygen transport, hemoglobin binding allows efficient oxygen delivery to transport and distribution, solubilization carries insoluble molecules, um, lipids and hormones in blood. Targeted delivery delivers substance to specific tissues or organs. Toxin remove, binding proteins help eliminate waste products. Therapeutic monitoring, protein levels matter, that means liver or kidney disease alter binding capacity. Monitoring needed. Measure free drug levels, not total concentration. Diagnostic application, biomarker detection, antibody antigen binding used in diagnostic tests. Research tool studying protein interaction levels, disease mechanism.